So it's a pleasure for me to introduce you to Dr. Oscar Perez Escobar, a friend from Lancaster Botanical Garden. Oops, I'm nervous. Sorry. Dr. Oscar Perez Escobar is a research group leader and Sainsbury Orchid Fellow at the Royal Botanic Gardens, Kew, from the United Kingdom. He received his PhD in evolutionary biology and plant systematics from Ludwig Maximilians Universität München. I hope I'm pronouncing that correctly. Maybe not. Specializing in orchid systematics, evolution, and genomics. Dr. Perez Escobar primary research centers on the phylogenomics and of Orchidaceae, linked to the plant and fungal trees of life strategic output. Utilizing advanced sequencing techniques to construct the orchid tree of life, Dr. Perez Escobar investigated the drivers behind orchid diversification across shallow and deep evolutionary time, including the impacts of biotic and abiotic factors and the biogeography of orchid colonization across terrestrial ecosystems on Earth. He is also interested in the effects of artificial selection on genomic architecture using comparative genomics on modern and ancient specimens. Additionally, he investigates the origin and evolution of cultivated plants utilizing archaeological remains to study plant genome dynamics in response to environmental changes and human interactions. Passionate about understanding plant evolution at both micro and macro scale. So, Dr. Perez Escobar, it's a pleasure to have you here.
Well, I cannot really express how grateful and privileged I feel to be here with you, sharing and learning. So my gratitude goes to the organizing committee for this invitation. I thought I could share with you today some examples from research at Kew on assembling and understanding the monocot trees of life. Right. So this is the outline of this presentation. Given the context of this meeting, I thought I could very briefly share a bit about the history of monocot research at Kew, very briefly. And then I would like to talk about 3 examples of Kew research in monocots, mostly the plant and fungal trees of life, some date palms, and orchids. Right. So Kew has a very long history, as you might know, of monocot research, which actually started with the very first directors that Kew had, with some orchid work, taxonomic work, and this reflects actually on the comprehensive collection we have at Kew, the monocot herbarium collection, and the oldest living orchid collection that was actually established somewhere in 1770 something. And what you can see actually here is a very cool plant specimen which was collected by Charles Darwin and then was named as Chlorea magellanica, I think, by Joseph Hooker, if I remember correctly. Right. Then moving forward some centuries after, I came across this photograph, and with apologies to some of you which are actually here photographed. This photograph is important because it was actually the photograph taken, with all the very first attendees of the very first monocot meeting ever, which was celebrated in Kew. And with all your permission, I would like to point out some individuals or colleagues. It has been a very exciting time to be alive there, like tremendous minds. We have here, I believe that is Professor Tom Givnish, but I might be wrong. I mean, I was like 5 years old when this photo was taken, so I've done a strong effort to like point faces. I think that here we have Sean Graham somewhere. Then there is our own Ana Maria Juliette from Brazil. There is Phil Crape, of course, and I think somewhere in the back, there is Peter Linnet. Those are the few colleagues I managed to identify. But then I pulled out this photograph because I wanted to highlight 2 colleagues here. We have others, of course, Richard Bellman, which I can forget. We have here Paula Rural and Sabo Mayo, who is, I think, not in this photograph. And the reason why I'm pinpointing this is because it was, in fact, Paula Rural and Simon Mayo who conceived this very same conference we are celebrating today, right? And the main purpose of this conference, as far as I understand, was actually to establish a network of collaboration in monocots research. And as a very young scientist, it really brings me joy to see how much fruit this has borne so far, right? Right. If you want to tag along here in this photograph, by all means. I hope I didn't forget anyone here present in the room, but apologies if I do. Right. So moving forward, perhaps one of the most, let's say, important projects that Q is working on at the moment, linked to monocots, is the plants and fungal trees of life. I think that Professor Tom Gimnish did a really good job, explaining what pathol is, so I'm not going to really dwell too much into this. But just to really simply summarize, the main goal that the plants and fungal trees of life has is to expand and populate the tree of life for plants, in our case, right? And the way we are doing this is very simple, which is basically sampling 1 or 2 representative species for every genus of accepted angiosperms. And we are doing this by sequencing the so-called angiosperms 353 genes, right? This is a massive undertaking, as you can imagine, which involves about 294 individuals in 117 institutions and 21 countries. So far from being one-side project, it's something that is rather like really cooperative effort. And some of the colleagues that you see here, they are part of what we call the core pathol team, which are responsible for producing debates, networking, doing some of the science, and so on. But this really goes well beyond Q, as you know. And as far as I understand from Bill Baker, so far, this collaborative effort has resulted in 44 publications since 2018, which is a tremendous and massive output, right? But moving now into more exciting news, which I think some of you might be aware, very pleased to share on behalf of my colleagues at Q the latest angiosperm tree of life that has been just accepted in Nature. This tree is, as far as I understand, the very first tree produced from nuclear genes, extensive nuclear genes, that represents all the 64 orders and 416 families. Forgive me if I forget some families here. And includes about 8,000 genera, so around 60% of the accepted angiosperm genera, which are around 8,000. And all the credit here goes, they are like about 300 authors, of course, but the credit goes here to Alessandro Sontini and Tom Carruthers, which are like the first co-authors of this publication, or soon-to-be publication, then led by William Baker as a senior and corresponding author. So I'm sure that all of you know Bill, but just to clarify, it's not the dog, but this gentleman there. Okay. Cool. Let's have a look. And of course, you can see monocots here, which is like a big part of the angiosperms. So let's have a very quick and brief look on this phylogeny. Right. So here what you see is a section of our new tree in comparison with the prevailing classification system, which is APG4. And as you can see, there are some differences in the placement, so we are just focusing on monocots. And if you want to really know more about this, just please get in touch with Bill Baker or the co-authors, the leading co-authors. Just to clarify, what you see here on the nodes is basically the support of this phylogeny. Whenever you don't see any node on any bipartition, means that we have full posterior probability support in an astral analysis, species tree analysis. And as you can see, when we compare the position of orders on like the monocot tree of life versus the APG4, there are like not really many changes, so it's like rather very similar phylogeny. And there are a number of nodes which still lack support, but I think that overall the support is very good. This phylogeny includes about 1,700 genera of the currently accepted monocot genera, so that's about 62%, if I understand correctly. And of course, all the families and all the orders that are currently accepted. Now, this diagram that you see here on the right side is basically a comparison between the position of families in this new phylogeny versus the latest plastid phylogeny of angiosperms. And again, like there are very few changes in the position of some lineages, mostly, I think, between Heliconiaceae and Musaceae. But again, this is a very similar phylogeny to like what we know so far, and based on the current phylogenies. And again, this is the work of my colleagues I was asked to share here. Right, but then, I mean, obviously, this panorama is very good. Like, I think that based on these phylogenies that have been produced so far, I think that we have a good understanding of how things are related to each other, save some relationships deep back in time. But that this doesn't really tell us how far along are we in understanding how monocot species are placed on the tree of life, right? So to answer this question, a few years ago, 2021, we did a survey across all seed plant families to try to understand how many of them have been sequenced or at least are represented in NCBI repositories by at least one of the 10 most commonly sequenced markers. So here in this diagram, which is a Smith and Brown phylogeny, sample to family level, and on the right side, what you see here, bar plots are the number of species for all families, showing only selected families. But you can see when you zoom in the monocots, that at least most of the monocot families have less than 50% of the species represented by at least one sequence in NCBI repositories, right? And one interesting pattern where here warm colors represent a lot of missing data, let's say few species sequenced, is that the largest families are the most underrepresented, and this is a pattern that is not new, right? So it seems that we still have a lot of work to do to try to place all monocot species in the tree of life. And by the way, all the data that I just presented that was used to build this tree is freely available here in the Tree of Life Explorer and NCBI as well, that anyone can query it. And if you go into the Explorer itself, you can see all voucher information, provenance, all of that is provided together with the protocols that explain how this data was generated and curated. So just to say that this is like a fully transparent process, right? Cool. I would like to talk a little bit about my own research I've been conducting with some colleagues. I'm actually glad I chose to talk a little bit about palms, following Maria Sanin's comments on the fact that palms have been a bit underrepresented here, right? So I'm sure that palms don't need introduction. As you know, palms are far from being a trivial family. They represent a source of food and fibers for indigenous communities, but also millions of dollars in economy on several crops, right? They are also a cornerstone species in multiple ecosystems. And I do personally like palms because from every now and so often I find very cool orchids growing on them, but that's it. This is just to say that by no means I am a palm expert, really. But then my interest on palms is because I could use them as a model to, let's say, learn about genomic techniques, something that I expressed to Jim Levensmark a few days ago. And I'm going to talk a bit today about a work we did with some colleagues on date palms and domestication. Date palms, or Phoenix dactylifera, is one of the oldest or earliest domesticated tree crops with the earliest records of utilization that go back about 4,000 years ago. The importance of date palms is actually reflected on all the rich archaeobotanical and archaeological records that go back thousands of years, which actually range from panels, stone panels, to char seeds, and even coin engravings that have hundreds to thousands of years. And an important thing of date palms is that they provided a suitable environment for the domestication of other crops because of the environment that they subsisted. Oases allow it to grow other plants under them. Right. So just to introduce a little bit Phoenix. Phoenix is a genus that has around 13 accepted species. We don't really know how many they are because there is a lack of a serious taxonomic treatment, which are distributed in Africa, Asia, and the Mediterranean. Something that I found very interesting when I started working on this group is that there is no solid phylogeny of Phoenix, despite the fact that it represents millions of dollars for several economies. And as I said, indigenous communities or some local people use them as a food source and source of materials as well, which is very interesting, right? I mean, I am an orchideot, so to speak, and I thought naively that, there is still a lot of work to do on plant groups that they don't represent much for economies, but in fact, when you go into cultivated plants, there is still a lot of things that we need to learn. What you see here is basically the most accepted relationships of Phoenix dactylifera with their closest or sister species. It is accepted that Phoenix sylvestris is like the sister species of Phoenix dactylifera, and Atlantica is basically a species that is restricted to Cape Verde, but thanks to genomic analysis and population genetic analysis, it is accepted that it's basically part of the same population structure, so to speak. What you can see here in this map is the distribution of these species that you see here. One important thing is that our current understanding suggests that Phoenix dactylifera is native or originated in Asia and then was subsequently introduced in North Africa for cultivation. And some of these native and cultivated populations overlap range with some other species, but definitely not others. In particular, Phoenix theophrastii, which is a palm that has basically no use so far. Right. I think one of the coolest pieces of research I have seen on date palms is the following one, which was done by Michael Puruganam and his team, who I could say they are like at the forefront of genomics on date palms. And what this publication presented actually was interaction or gene flow between Phoenix dactylifera and Phoenix theophrastii, which, as I said before, is basically a palm that doesn't share distribution range, like completely with cultivated dates or Phoenix dactylifera. And what this colleague found is that actually about 10% of the genome of Phoenix theophrastii is shared with Phoenix dactylifera, right? They managed to pinpoint which portion of the genome of Phoenix dactylifera is shared with theophrastii. But then one key question that remained outstanding from this work is the timings of those interactions, right? When it happened in time. And then the goal of that project was, or of our project was to answer this question. We wanted to understand or discern the timings of this genetic exchange of Phoenix dactylifera and theophrastii and possibly other species. So how did we address this question? We basically queried gene data or genomic data from NCBI repositories, mostly from date palm, Phoenix dactylifera, plus the close living relatives, which are these 4 species I introduced before. But we also did something innovative, which was basically sample or try to sequence the genome of a piece of date palm that has thousands of years, that is thousands of years old. And this is because these pieces of plants is like, timestamps, like say, a piece of evidence of genotypes that perhaps might be extinct and that are like thousands or hundreds of years old, and can perhaps help us informing where in time these gene flow events happened. Then we could do a series of analyses to understand the ancestry and relatedness of this sample, this ancient sample, versus modern samples. And then we produced a chromosome-level phylogeny in conjunction with molecular clocks and multi-species quality analysis to try to date these gene flow events. Right. This is the object that we analyzed in our study. And actually, this object itself has a really fun story. This object belongs to the Economic Botanic Collection, and it was found in the 40s, I think, in the ancient necropolis of Saqqara. I will talk a little bit about that in a minute. But the story of this object is interesting because the colleagues that found it in these excavations in Saqqara thought that at first this was like a headpad, basically kind of an item made of date palm leaves that some people in the past were used to rest their heads. And we went with that, those data, and we brought our manuscript with that assumption that this was actually a headpad. But then Mark Nesbitt, who is an ethnobotanist and the head of the collection at Kew, had some friends in the British Museum, and by sheer chance, they found an item which was very, very similar, which is the one shown here in this photo. This item actually had better notes about the utility of this sample, the purpose of this item. And it turned out to be something completely different. It was basically a seal that Egyptians used to use to seal jars where they were storing beer or grain, et cetera, right? So very, very different purpose. But then in order to really assure that this is an ancient item, what we did is that we radiocarbon dated it, just to be sure, because sometimes mistakes can happen. And what came out of the analysis is that this sample is actually about 2,100 years old, and confirming that, in fact, it's an ancient sample, right? That is, something that we can use to derive ancient DNA. And the plot that shows here provides the evidence of the age of that sample. Now, the place where this sample was found is very cool. is known as the animal necropolis of Saqqara, which is located somewhere here, 20 kilometers south of El Cairo, and it's an UNESCO site, and it's mostly known, I think, by the millions of mummified animals that you can find in there. And what you can see here is actually a pyramid of Djoser, which is like the very first pyramid built, as far as I understand. I think that one cool thing of like doing research in something else than orchids is that one can manage to justify trips to go and see things. I could hardly say to Alex, please support a trip to go and look for orchids in Saqqara, right? Right. So as you can imagine, sequencing the DNA of items this old is extremely difficult. And most of the times, like, the success is very, very small. Of all the data you produce, sometimes you will get 1 to 10%, if you are lucky, of endogenous DNA. That is DNA that actually matches the organisms of interest, and not just what we call exogenous DNA or contamination, which can actually be human DNA, bacterial DNA, fungal DNA, et cetera, et cetera. So one way to do this is by mapping all this data that we produce against a reference genome, which we have, and then really see whether these ancient samples, this ancient DNA, has a unique trace of damage that only samples like historical and ancient DNA have, which is basically what we call a deamination, or misincorporation of nucleotides that tend to occur towards the end of the fragments. So this analysis that you see here is basically a comparison of these misincorporations or artificial mutations that occur in modern samples versus ancient samples. And as you can see here, our item sample, ancient item, whenever you try to model these misincorporations, you can see that there is an elevation of C2T's misincorporations toward the ends of the rig fragments, which is what is shown here on this axis, and this is the frequency of this misincorporation. And so this can really reassure us that what we are sequencing is ancient DNA and not just like cross-contaminations of modern day palms. You can see the difference here with the modern sample. And another key signature of damage is the frequencies of the fragments, right? Ancient DNA or historical DNA tend to be super short. I don't know you, but I find this really, really fascinating, the fact that we can now, thanks to technology, we can really access the DNA of these things. It's just really mind-blowing. And by the way, all of this work was done by Professor Michael Freiter from the University of Potsdam. He's a really renowned figure on ancient DNA works on mammals mostly. Right. So let's see what we managed to learn from this sample. What we did first was to try to understand how this ancient sample is placed among a panel of modern accessions. So we built a plastid and nuclear phylogenies, or let's say networks. And what we found out is that our sample, which is called Saqqara, is actually nested among accessions of African cultivars and what we call Phoenix atlantica, which, as I explained before, is just another date, but with a different name, right? Something very interesting that you can see here is that there's actually a chloroplast capture event of Phoenix sylvestris, which is the sister species of dates, cultivated dates, which is distributed here and shares natural range there. And here we have mostly Asian accessions of Phoenix dactylifera. Now, this chloroplast capture event is understood and can actually inform possible gene flow events from Phoenix dactylifera towards sylvestris. And what you can see here on the right is basically a network inferred from SNPs, nuclear SNPs, which actually show and confirm that what we sequence is, in fact, Phoenix dactylifera and not any other species, right? And as you can see, there is a clear cluster here of Phoenix dactylifera where Saqqara is nested. We have here sylvestris, which is placed as sister to this cluster, and then they are, let's say, related, closely related species. Then to try to understand how, to try to trace the ancestry of this genome of this ancient sample, we conducted population structure and PC analysis. And when we assume 4 to 5 ancestral populations, which is like the best fit for our data, we can actually see that most of the genome of the Saqqara sample, which is shown here, can be traced to African dates and dactylifera. This actually here is mostly Asian date palms. And when you look at K3, you can actually manage to trace some of the genome to Phoenix theophrasti, which can perhaps provide an evidence that by 2,000 years, there was already some gene flow between Phoenix theophrasti and date palms, right? And what you can see here on this PCA is basically the position on, or how this ancient sample is related versus other date palms, which is congruent with the structural analysis. So basically, in short, the point with this slide is to say that the genome of our ancient sample is let's say, more similar or can be traced to North African dates than any other date plant group or any other species. So now to properly conduct test of gene flow, what we did was to do something what we call a BABAB analysis. This is a bit of a thing hard to explain, but it's simply a way to try to understand how ancestral or derived alleles, as defined by an outgroup sequence, deviate from the assumption of species trees. So here in these bar plots, blue represents BABA alleles and orange represent ABA alleles. And what we'd expect, like under a species tree relationship, is that our sequential sample will share more alleles, derived alleles, with Dactylifera. But in this particular case, what we can see here with the statistical support is that there are more alleles shared with our ancient sample and Silvestris, then providing evidence for gene flow event, not only with Theophrastii, but Silvestris. And here in this plot, what we can see here is evidence of statistically supported evidence of our ancient sample sharing alleles with Theophrastii, right? Interestingly, our results were also confirmed by a paper that was almost published at the same time than ours. This paper is super cool because basically what these colleagues did was to germinate seeds, date-palm seeds found in southern Levant that were 2,000 years old, which is completely crazy, right? I never thought that such thing could be possible, but they did it. So they did the same analysis than us. They germinated the seeds. They sequenced the leaf of these 2,000-year-old seeds. And they found the same gene flow patterns that we discovered, basically. Right, but then, let me go back. While these analyses tell us that gene flow event between Theophrastii and Silvestris existed or occurred at least 2,000 years, which is the age of our sample, it doesn't really tell us where there was evidence for ancient gene flow, right? Something that predates the age of our sample. So to address this question, what we did first was to build a chromosomal level phylogeny using a pseudo-haplotype approach, which is basically produce consensus sequences across chromosomes for the samples that we had, and we mapped against that reference for all the species that we analyze. And this is only possible because in Phoenix, the architecture of the genome is more or less conserved, and you have the same chromosome numbers, which I think are 21. And we have a very good genome of reference with high contiguity, right? Sorry. And then what we did is, using molecular clock analysis in conjunction with multi-species coalescence methods, which allows you to use population circumscriptions to say individuals belong to species A, B, and C, or populations A, B, and C. We estimated ages of divergence on the species tree, which is what you see here in blue. This supports our understanding of relationships of Dactylifera with Silvestris and Theophrastii, where Silvestris is the sister species of cultivated dates. But then as we did the analysis, we actually found two other dominant topologies, right? So, which actually showed the age of gene flow between African dates and Theophrastii. And then Theophrastii itself with both populations of Asian and African dates. So this actually provides evidence that we didn't only have evidence of gene flow that is very recent, at least occurring at 2,000 years old, but also something that goes back to at least 7 million years ago. Now, you might say that this perhaps could be the effect of incomplete lineage sorting and so on. But then when you assume a model of incomplete lineage sorting, you would tend to see one dominant topology, and then any other possible topology will occur in equal frequencies. But what we did was actually to calculate the frequencies of these three topologies plus any other possible topology. And what we discovered actually is that there is one main topology occurring in high frequency. And then, oh, I think Stephen is falling to sleep. Sorry, it's not orchids, but orchids are coming soon. Just bear with me a minute and you will hear about orchids. And then what you can see here is that there are two other dominant topologies that occur in less frequencies, and then any other possible topology is shown here in gray, then suggesting that these topologies we are seeing here are actually derived from gene flow and not ILS. Right, now moving to orchids, which is what I'm being paid for by Q, right, to do research. These dates and other crops is stuff I do on my free time. Like, I'm not joking. Well, I'm sure that orchids don't need interaction, right? And they are far from being a trivial group because they represent millions of dollars in the economy, and also they are the source of food and fibers for people. Something that I find very interesting of orchids is like the wide distribution they have, nearly worldwide, but also how they have become dominant in some of the floras worldwide, especially in the tropics. So I have picked two examples here, right? So we have here New Guinea, and this study was done by a colleague, Rodrigo Camaraleret. And when you try to just dissect the diversity of plant families in New Guinea, you tend to find that orchids are like the most diverse component, like the most species-rich component. You tend to see the same kind of trend in Northern Andes, and whenever you calculate the species diversity across an elevational gradient, what you see that actually orchids, they tend to come first in terms of species numbers, right? So something I'm very interested in is try to understand how these floras assemble worldwide. And orchids obviously are a great example, given the wide distribution they have and the fact that they are very rich, or species rich in some environments. Now, as you might know, and as Professor Tom Givnish explained, there has been hundreds of phylogenies published, so our study actually stands on the shoulder of giants, right? Most of these phylogenies, of course, are focused on specific groups. Some others, there are a few, have used hundreds of genes, either from plastic genomes or nuclear genomes, and are focused towards the entire family. And then one work I would like to highlight is the work of Professor Givnish and collaborators, which was actually seminal, and we used as an inspiration to build our own, right? Who was the one who actually conducted formal phylogenetic comparative methods on biogeography and diversification using a plastic phylogeny. So the goal of our project is the same as BAFTA, right? We want to build the most complete and robust phylogenetic framework of the orchid family. And to do this, we want to sample all genera that are currently accepted, which are around 720, using the Angiosperm 353 kit. And then something that we want to do to enable fine analysis to geographical scales and to basically make more robust inference on biogeography and trait evolution is to try to combine, or try to, let's say, expand as much as possible all the species sampling, right? And in my opinion, or in our opinion, the only way to do this is by combining whatever is available in any public repository, for example, Sanger sequences, and merge that with this framework we are constructing, right? And we think that with this framework, we can perhaps, as outcomes, understand changes in speciation rates through geography in detail, revisit some hypotheses on historical biogeography, and then one personal interest is more, try to understand more, the mode and tempo of origin of modern orchid floras worldwide. So the very first paper that came out from Q and orchids is the one which I think Tom explained very well. We here presented the first orchid phylogeny from hundreds of nuclear genes, where we sampled 69 genera, which are about 99% of the accepted genera. This paper had actually a very good impact in the scientific community and was actually featured as the most read from 2021 to 2023 in American Journal of Botany. And something that was cool of this paper is that we conducted the first formal analysis on how the nuclear phylogenies are different from the plastic phylogenies. But not just to say as to, oh, well, position A is different here in the nuclear tree than here in the plastic tree. We actually tried to quantify the magnitude of the incongruence. And to do this, we used a tool that we designed during my PhD with some collaborators. And what we found out is that actually phylogenies of nuclear and plastic genes, they have different levels of incongruence. They are mostly congruent, of course, but they have some differences at different phylogenetic levels, right? So we did this not as to say which is the right or the wrong tree, because this is not the whole point of this. I mean, as an evolutionary biologist, I'm really interested in trying to understand how life originated on Earth. And obviously, incongruence is part of that process. So we don't want to cherry pick, oh, nuclear tree is good, plastic tree is bad, because it just does not make sense in my very humble opinion, right? So moving on to our latest outcome, which I'm sure you might have seen, we have presented a brand new phylogeny, which actually includes about 40% of the sequence genera, well, to be precise, 38% of the accepted genera, 90% of the tribes, and 81% of the sequence subtribes. What we can learn from this analysis, which is a super tree network, is that the incongruence between the nuclear genes is very low, as judged by normalized Q-scores. But then when you dissect the support of concatenated analysis, you can see that most of the bipartitions fall between intervals of 70 to 100. And when you do the same, when you calculate the proportion of support across individual gene trees, you can see that the great majority, or a big part of these bipartitions, fall in these intervals. And as expected for gene trees, there are also many that have like low support, but that's okay, right? I mean, we cannot expect every gene tree to have 100% support because that's never going to happen. And just to remind that these analyses that we are going to do here, they are meant to be used to infer a species tree. Something very cool of our studies that we managed to place for the first time some genera that were never sequenced before. One example is Claderia, which was placed by Mark Chase and collaborators in Eulofini based on morphological similarities. When we sequenced this Claderia, which we actually selected from type specimen, and at that time was a monotypic genus, was actually placed as sister to, or nested in Agrostophyllini, right? Or at least sister to all Agrostophyllini that we have sampled so far. These results were actually later confirmed in a publication that came out just before ours, using whole CDS, and they managed to attain the same results. I think that that sample is, where is it? Yeah, so here. You can see here 2 samples of Claderia plus a new species they describe, also in Agrostophyllini, right? So then confirming our results. But then something that we wanted to do is to, as I said, enable fine, let's say, detailed analysis on speciation rates and biogeography conducted to geographical scales. And to expand our taxonomic sampling, let's say that the only chance or the only choice we had was to try to make use of everything that is available in NCBI, namely Sanger sequences, and try to merge that in a smart way with the NGS backbone that we have produced. Then, inspired on a publication by a colleague, Nicolas Chassot, on butterflies on Nature Communications 2021, I think, we built this kind of pipeline, which basically departs from dating independently the nuclear angiosperm 353 datasets and the Sanger datasets. So departing from this common background of having ultraviolet trees dated in BEAST, we can then prune and graft phylogenies from the Sanger datasets into the NGS datasets, then having density sample phylogenies. We did this because, I mean, we know that uncertainty on these datasets is huge, right? Especially given that some relationships are not well understood, and also because there is, variation along branch lengths and therefore ages estimates. So if we rely on a series of phylogenies for all our analysis instead of just one, I feel that we can perhaps account best for phylogenetic uncertainty, right? And this is exactly what we did. We ended up having a set of 10 trees which we drawn from our BEAST analysis, which were then merged into what we call super trees. And we used these 10 trees to all our analysis. So to put these trees to good use, we inferred biogeographic analysis using a DEC model optimized in C++, which is basically a good way to conduct analysis in thousands of species. So our data set basically included nearly 2,000 species, which is like the largest data set of the largest orchid phylogeny ever assembled. And what our biogeographical estimations revealed is that actually orchids originated in Laurasia about 80 million years ago. So we followed the same calibrations that Tom Gimlish and colleagues used. And something very interesting that actually we found is that on all these 10 reconstructions we did, independent reconstructions, about 8 of them support this Laurasian scenario. About 2 of them represent different scenarios. So actually this tells you that uncertainty matters, right? So instead of working with phylogeny, I would really like to encourage the use of multiple phylogenies and try to see how much variation in branches and positions affect reconstructions. So this Laurasian scenario of origin of orchids is supported by the origin of other monocot groups, for example, palms and Dioscorea. What you see here is a fossil of Palmoxylon, which is like widely distributed fossil in different parts of the world, some of which have been found in North America and are about 80 million years. And the last thing we did, just to finish, was to try to understand correlation patterns between speciation rates and species richness in geography, right? Which was done in conjunction by compiling geographical records, distribution records from GBIF. And what we found are some very interesting patterns, which are basically that in some areas you have a direct relation between species diversity and high speciation rates. So that is the case of tropical Andes and Central America. But in some other cases, you see that you have huge diversity of species, like species richness in Australia, but then it seems that that diversity took longer to accumulate than expected. You can access our paper online. I'm very pleased to share that, despite the fact that this paper has been published just 2 weeks ago, is now one of the most read of all published papers in phytologist. I'm also very happy to share with you that we made the cover. And yeah, we are. So this is just to highlight that we are really a huge team, as you can see, all the co-authors. We are really open to cooperate. We're actually looking for new collaborations to start assembling the third version of this phylogeny. So if you want to get involved, just like really get in touch with us. We are happy to work. And I think that the purpose of this very same meeting is, in fact, to network and make science together. So just really get in touch. Lastly, how much time I have got? Fine, okay. So these are just really simple conclusions of my presentation, right? As echoing the words of many of you, we really need a robust and density sample phylogeny to unravel the mode and tempo of evolution of monocots. And to me, given that, despite the fact that it's cheap to sequence now via NGS many samples, we cannot really afford sequencing thousands of species. And in my humble opinion, perhaps one approach is try to combine whatever is out there with what we are producing in smart ways. So perhaps supertrees approach are one way. I don't know. This is really open to discussion. There's actually a lot of exciting progress on monocot relationships inferred from nuclear datasets, which are mostly congruent with plastic datasets. And that, yeah, we still have a really long way to try to understand how monocot species are placed on the monocot tree of life. Lastly, of course, none of this research that I shared today, especially the one I have been conducting with my colleagues, would have been possible. So I am very privileged, and it's an honor to work with all of these collaborators. I would like to highlight the role of these gentlemen, of these people here shown in this photo, some of which sadly couldn't be here today. These are friends and colleagues and mentors, and without them, I wouldn't be here, really. And thank you for your attention.
Not yet. Oh, thank you, Oscar, for that wonderful talk. So we're gonna open a period for questions.
Thank you, Oscar. Congratulations for nice work and very interesting seminar. I wanted to ask you regarding the date palm. You have very nice results regarding introgression from different species into the cultivated date palm. Do you happen to know which regions are affected by this introgression and which genes?
Yeah, that was actually found on, well, okay, so if you are asking in terms of like our ancient DNA sample, not really, because what we record was like rather few, SNPs, but then if it's about this paper, I don't know. So this is because actually this work was built using a reference genome, which is highly contiguous, but then the pseudomolecules represents just 50% of the entire genome, then all the rest is placed in scaffolds. But yeah, I don't think that's like really fully understood. You are Ovidio Pound, by the way, right?
Yeah.
It's great to meet you. I'm a huge fan of your work. I would like to talk to you at some
Thank
point.
you.
Anybody else?
Great talk, Oscar. Thank you very much. I have one question. When you show this map with the Andean countries and they have some numbers, what are those numbers? When you were starting talking about the orchids.
Okay. Sorry, it's a bit slow. Here.
Yeah, those ones. Yeah,
These, sorry, these numbers.
Yeah.
So these are basically Andean species diversity, regardless of the family.
Okay.
So
okay.
yeah,
Thank
but
you.
then what is interesting on this plot is the number of species of orchids and let's say top 5 or 10 families across different elevational gradients. And as you can see, from 100 to I think 3,000 meters, orchids are like the most species-rich family.
Okay. Well, thank you very much.
Pleasure.
So really great talk, Oscar. Very nice.
Thank you.
I had just a couple of points. One was, I agree with you that bringing in other data, other loci, is very important in terms of extending coverage, and we did that as well with our plastome phylogeny. But I do think in our case, a lot of the branches were supported only by 3 genes, and we never highlighted those specific. I mean, it was obviously in the appendices, and you folks don't really highlight the branches that are only due to sort of recruiting or downloading data from other things. And I think maybe for the future, we should all, when we're graphing the results, just highlight the branches that are really due to low amounts of data. I think that would really help things. And then I guess the question I had was, you used a super tree approach, and a number of people have argued on theoretical grounds that super matrix approaches are better in terms of recovering phylogeny. I don't really care, frankly. I mean, I'm not an agent for either side, but I'm just wondering, did you folks try a super matrix approach, and if so, what results did you get?
Well, thank you, Tom, for your question. I think that pursuing a supermetrics approach here is prohibited simply because you don't have compatibility between the data sets. And even so, if you manage at some point to, let's say, data mine ITS, MATK, like the most commonly sequenced markers from this data, if you try to align the BAF2 genes plus those Sanger genes, and then if you include thousands of samples in the supermatrix, you will have a lot of data missingness, and that definitely, 100% sure, is going to bias your analysis, you see. Where it's like, phylograms or ultrametric estimation, and BIST particularly is very sensitive to that. So I wish there would be a way to follow a supermatric approach, but I think it's rather difficult, really. Thank you.
One last question.
First of all, don't do supermatrix. I could talk with folks about that during coffee hours. Secondly, what groups are diversifying most recently? Which ones are actively radiating? And how is habitat destruction and other conservation issues that we're experiencing in the Anthropocene influencing diversification of some of those groups? Increasing diversification or decreasing diversification? And your ideas on this. I know you don't have data, but your ideas on these.
Well, I mean, I wish we could answer those questions with the data like we have, and it would be actually something very cool that one could do, right? I can only say about like the groups that, let's say, have the highest speciation rates, as in terms of modern speciation rates. And by the way, this is an approach that we choose to follow instead of like trying to understand changes of diversification rates back in time. Because, I mean, as you know, this is really hard to estimate. We have like a really relatively low sample. This is, I mean, it's 2,000 species, but it's just 9% of the entire family, right? So it's still like really undersampled. So we decided to work on modern speciation rates, which, as far as I understand from Daniel Rabowski, they are less prone to error. But we can see here, I don't have the slide here, but some of the groups that has the highest speciation rates are Protalini. These are congruence results based on Tom Kirthy's results as well, and colleagues. As we know, these are basically nootropical groups, which are basically distributed in Central America and South America mostly, mostly mountainous regions. But my colleagues from Lancaster can talk more about Protalini. But it would be really, really cool if we could try to, like, join or try to link how deforestation is affecting speciation rates. Something that we can actually say is that based on this analysis, we could try to find areas that have more or higher evolutionary potential, as in areas that have higher potential to produce more new species, and perhaps try to conserve more or inform conservation priorities with this information. But that's all I can say for now. Thank you for your question.
Well, thank you very much, Oscar, for that wonderful presentation. Let's give a round of applause for Oscar. And